Throughout history, few events have left as indelible a mark as the Black Death. But could the key to its prolonged devastation lie within a single gene of a microscopic organism? In the annals of human history, pandemics have reshaped societies, economies, and the very fabric of civilizations. Among these, the plague stands out, not just for its immediate impact, but for its persistence across centuries. Now, new research reveals that a single gene in the plague bacterium, Yersinia pestis, may have been the reason this killer never fully disappeared. That gene, small, ancient, and deadly, played a role not just in how the bacterium attacked the body, but in how it etched its presence into the DNA of history itself. The Black Death of the 14th century decimated populations, claiming the lives of an estimated 30 to 50% of Europe's inhabitants. In cities like Florence, London, and Paris, entire neighborhoods were wiped out in weeks. Cathedrals filled with corpses, laws collapsed. Survivors turned to mysticism and violence for answers. But this was just one wave. The plague had struck before, during the 6th century with the plague of Justinian, and it returned repeatedly through the centuries, often in smaller but still deadly surges. These recurrences were not isolated, rather they were connected by a hidden thread: the bacteria's capacity to endure, mutate, and circulate, sometimes silently. Across Europe, Asia, and parts of Africa, plague outbreaks shaped migration, revolutionized urban planning, and even altered military campaigns. In China, it contributed to the fall of the Yuan dynasty. In the Ottoman Empire, it disrupted trade along key Silk Road corridors. It wasn't just a European event, it was a global one, rooted in microbiology and amplified by human movement. Some societies never fully recovered. Entire villages vanished from maps. Survivors passed on generational trauma, adapting religious rituals and burial customs to account for mass death. The long tail of the plague's psychological and cultural legacy endures even now. A team of researchers, led by evolutionary geneticist Hendrik Poignard at McMaster University in Canada and collaborators from the Institut Pasteur in France, published a groundbreaking study in Science on May 29, 2025. Their work zeroes in on a gene within the Yersinia pestis genome, PLA, short for plasminogen activator. This gene allows the bacterium to break down barriers in human tissue and spread rapidly, especially through the lymphatic system and bloodstream. It is directly involved in causing both bubonic and pneumonic forms of plague, each one a lethal manifestation of the same organism exploiting different vulnerabilities in its host. PLA is a molecular key, unlocking the host's defenses and enabling the bacterium to bypass immune checkpoints. It doesn't just facilitate spread, it alters the balance between host and invader, tipping the scale toward catastrophic infection. But what made this discovery so impactful was not just the gene's presence, but the number of times it was duplicated. Through the analysis of ancient genomes from plague victims across Eurasia, extracted from teeth and bone, the researchers found that strains of Yersinia pestis with multiple copies of the PLA gene were vastly more virulent, often killing the host in a matter of days. Other strains with fewer copies had lower fatality rates, but infected hosts for longer, increasing their potential to spread via fleas or human contact. This duality between quick lethality and lingering transmission offered the bacterium a powerful evolutionary tool. It could adapt its virulence to the context: cities, trade ports, battlefields, or remote villages. Sometimes it moved quickly, wiping out towns. Other times it moved quietly, embedding itself in rodent populations or spreading through asymptomatic hosts. The PLA gene wasn't just a weapon, it was a throttle, a biological dial that allowed Yersinia pestis to fine-tune its aggression in response to ecological pressure. High play copy strains were devastating in dense urban centers, where quick spread and short incubation favored hypervirulence. But in rural or less mobile communities, subtle strains persisted longer and traveled farther. This balancing act likely allowed Yersinia pestis to remain active for centuries without exhausting its host populations. It was an evolutionary masterpiece of disease sustainability. The long persistence of the plague can't be explained by human infection alone. Rodents, especially black rats, were the primary reservoir, and fleas the transmission vector. But the bacterium's success in these hosts required a different strategy. Too deadly, and the rodent dies before fleas can transmit the pathogen. Too weak, and the host's immune system clears it. By maintaining an optimal number of plague copies, Yersinia pestis could keep the rodent alive just long enough for fleas to feed, multiply, and jump to the next target, whether animal or human. This equilibrium allowed the bacteria to lie dormant for years or decades before re-emerging. Even today, plague endemic zones in places like Madagascar, Mongolia, and the southwestern United States are monitored for outbreaks in wild rodent colonies. Modern flare-ups, though rare, underscore the bacterium's tenacity and its ancient partnership with fleas and mammals. And the cycle was global. Maritime trade ships became floating plague engines. Infected fleas could survive on grain sacks, fabrics, or livestock. Entire economies unwittingly helped the pathogen travel continent to continent. Cargo routes became disease corridors, and no border, empire, or wall could contain it once the host flea system took hold. Hendrik Poinar and his team at the McMaster Ancient DNA Center are at the forefront of ancient pathogen research. Their work requires pristine conditions, specialized clean rooms, ultra-cold storage, and sophisticated sequencing technology. Extracting DNA from ancient remains is like reconstructing a manuscript from burned and scattered letters. But thanks to advances in high-throughput sequencing and molecular forensics, researchers can now sequence even fragmented, centuries-old genomes. The implications are profound. Ancient pathogens can be compared to their modern descendants, revealing how genes like plague duplicated, mutated, or vanished. We are no longer speculating. We are reconstructing microbial history, genome by genome. Their findings don't just explain past pandemics, they create a genetic map of risk. By identifying how virulence evolves, scientists can scan modern pathogens for similar gene amplifications that might signal a future outbreak. This isn't just about understanding history. It's about using the past to protect the future. Understanding how pathogens evolve, how they balance lethality and longevity, helps researchers anticipate what modern viruses or bacteria might do next. Could modern pathogens rediscover similar gene amplification strategies? Could we detect the rise of virulence early enough to intervene? Already, scientists are watching patterns in tuberculosis, cholera, and even coronaviruses, looking for telltale signs of genetic arms races. The lessons learned from a medieval disease are now helping us prepare for futuristic ones. Because while we've advanced in vaccines, diagnostics, and treatments, the core evolutionary pressures that drive pathogens remain unchanged. This isn't theory, it's preparation. The plague isn't just a horror of the Middle Ages, it's a genetic story. A story where one gene, plague, helped an invisible killer balance lethality with longevity. A gene that let the disease smolder rather than explode. A gene that changed the rhythm of history. Its story reminds us that the past isn't dead. It lives in our DNA, our medicine, and our pathogens. It endures, adapts, and evolves just as it always has. So the next time you hear about an ancient pandemic, ask yourself, was it fate or was it evolution written in the code of life? If a single gene can echo through centuries, what might be written in the genomes of today's silent threats? Stay curious, stay questioning, and subscribe to uncover more stories that science is only just beginning to tell.